Hi, I'm Roberto Barba and I'm going to present about PCR. PCR is short for polymerase chain reaction. This tool helps us to copy a specific sequence of our DNA. This can help us to diagnose some diseases, identify bacteria and viruses, and even match criminals to some crime scenes. And this process can almost get 100 billion copies of a DNA sequence in just a couple of hours. So, in general terms, this is a really simple process. And the first thing we would need is our PCR tester for us to get our materials and substances together. Then we will need our simple reagents. That would be our enzymes, primers, and some other substances that we will need. Then we will need a source of heat. This could help us to lower or increase the temperature of our substance. And finally, we would need a DNA structure. For us to get a DNA structure, we would take it from a cell, maybe from our skin, our saliva, our hair, or even from our blood. Now we're going to see what do we need to put in our PCR tester. There are in a total of 5 things we will need to use in our PCR tester. The first 2 things are really important because this will help us determine the start and the end of the DNA sequence we will need to use or copy. Second, we will need the DNA sequence. So we need a hair, we need saliva, we need blood, or we need a part of our skin. The important thing is we need a new DNA or living cells that has this DNA. If not, we can't use the DNA that has been in our skin, for example, for a long time. So we need living cells that have that DNA. Then we will need the nitrogenous bases that would be, for example, C, A, T, and G. These are the nitrogenous bases that we have in our own body and DNA. Then we will also need a source of heat. This will help us regulate the temperature of the process. Finally, we will need a specific enzyme that we call DNA polymerase. This enzyme will help us to create the new copies of our sequence of DNA that we need to copy. Now that we have everything we need in our PCR tester, we are going to go inside and look what is happening on the DNA sequence that we are trying to copy. So we have our DNA sequence, many nitrogenous bases. We have this, the primer 1 and the primer 2, and we have some DNA polymerase. What we know about primer 1 and primer 2 is that they are short pieces of DNA that are made in a specific laboratory. Primers can have any sequence of nucleotides you'd like. So in PCR there are 2 primers that are designed to match the segment of DNA you want to copy. So the first thing we will do is increase the temperature of the PCR up to 95 degrees Celsius. What will start to happen is that our DNA will start to separate itself from its helix structure and will create 2 straight lines of DNA sequence. So our DNA would be like this, no? And here would be our nitrogenous bases in different order and sequence. What will start to happen after this is that we will need to lower again the temperature up to 50 degrees Celsius. What this will create is that our DNA structure will try to get to a helix structure again, but we won't allow that to happen because the primers that we discussed are going to bond into our DNA structure. And what will happen is that one primer attaches to the stop strand at one end of your segment. The other primer will attach to the other end of the strand. So we'll have two primers that will define where does our copying sequence will start and when it will end. Once the DNA polymerase is able to cross the entire DNA sequence we are trying to copy, we will, instead of two structures, we'll have up until 4 different structures of DNA that we didn't have before. So we'll have now 4 straight lines of DNA sequence that in which we have the information or the DNA structure we need to copy. So what will happen next is that we will increase and decrease the temperature as many times as we want until this process is repeated a lot of times. So when we have 4, if we repeat this process again, we will have maybe 8 or more sequences, because these ones created new copies of itself in just a very little period of time. So we will start to have lots of sequences replicating themselves just because the increasing and lowering temperature and just with the materials and substances we put in our testing tube. So at a certain point, if we do 30 cycles of this process of increasing the temperature, then lowering the temperature, and after it all again increasing the temperature, we will have up until billions and billions of our desired product that will be the sequence that are purely the ones that have the information we want and are not bonded to the other sequences of DNA. So these ones that are the ones that are our desired product are only the copies of the specific section we need. So with this process, it's really easy to just copy a sequence of DNA when you need it, and in just a little period of time you can have billions of desired products and almost up to 60 of our original DNA sequences. So these desired products are the ones that we are going to use to define or categorize some new diseases, or to see a person that has a virus, or to diagnose it with a certain disease. And it will also maybe help us to see the information of a DNA and maybe match it with a crime scene that we need to know who the person was in that crime scene. So this is a really easy process in which we could know a lot of information, have a lot of information, and copy that same information as many times as we want. So this is PCR, polymerase chain reaction. Thank you. Once we know that DNA polymerase is used in this process, we need to understand that the specific DNA polymerase we use in this process is not the one that we use or have in our own body, because this specific DNA polymerase breaks down at a 95 degrees Celsius temperature, which is the temperature we need for the DNA structure to open. So the specific type of DNA polymerase we use is from a strain of bacteria that it's called Thermus aquaticus. This type of DNA polymerase works and survives in a 95 degrees Celsius temperature, and can also survive to a 72 degrees Celsius temperature. So it's the temperature we need for our process to be done correctly, and also for our DNA polymerase to still function. After the two primers attached to the DNA sequence we want to copy, we will need the DNA polymerase. The DNA polymerase is an enzyme that we have in our own bodies, and it functions to create and copy new DNA sequences for the production of proteins, or just creating new cells and the division of cells. In this case, we'll use DNA polymerase to copy the primers that we put in this section. So with the primers standing here, what the DNA polymerase is that it will go here and here. So it will advance one in this way and one in this way. So when it advances, it creates new nucleotides that will attach to the other bases that were in our DNA. So where does this takes the other nitrogenous bases? Well, is the one that we put in our PCR test tube. So these nitrogenous bases are going to go here, 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 and here after the DNA polymerase starts to create a new sequence of DNA.